Hey everyone, thanks for coming to this week's video lecture. Today we're going to start in on Unit 2's Chapter 8 on metabolism and respiration and fermentation. We're going to go over different types of metabolic pathways where we can build up different molecules with anabolism, or we can break molecules down with catabolism. We're going to talk about how enzymes make these anabolic and catabolic reactions more favorable. And we're going to see how they speed up reactions. We're going to talk about how ADP is a really important molecule at facilitating these metabolic pathways and helps do molecular work within the cell. Then finally, we're going to talk about different kinds of respiration and fermentation. And so with that, let's get started in chapter 8, where metabolism is the culmination of all of the different chemical reactions that happen within the cell to maintain homeostasis and keep the cell living. And these metabolic pathways and these chemical reactions are not just like one or two. There's actually thousands of these chemical reactions that are all networked together in a series of very convoluted feedback loops and regulatory pathways that respond to the external environment and the internal environment of the cell to keep the cell living. But we can classify these metabolic pathways or these chemical reactions in a couple of different ways. We can classify them as catabolic if we're breaking a big molecule into smaller components, or we can do anabolism, which is building up those smaller components into a bigger molecule. And so a lot of the times, we need to do both of these for a specific molecule because we don't always need specific things. Maybe we need a specific compound at a specific time in development. And so we will build up that compound at a specific time point in development, but eventually we'll need to break that compound down. And we might have an anabolic pathway to build it up, and then we have a catabolic pathway to break it down. And this is to help regulate levels of different chemicals and molecules within the cell to make sure that nothing gets overly over concentrated or saturated and could potentially cause harm. And so these chemical reactions sometimes happen spontaneously. Chemical reactions typically go from places of high energy to low energy spontaneously. And sometimes they still need a little bit of a kickstart of energy to actually get that reaction going. But we'll talk about that a little bit we'll talk a little about that in a bit. And then these chemical reactions sometimes, while spontaneous, and they just can happen on their own randomly, they don't happen very fast. And so, for instance, protein degradation can take hundreds of thousands of years to take a folded protein and break it down into its component amino acids. But instead, we could take peptidases or proteases and break that protein down in minutes. And so these enzymes are what are known as catalyses, and they speed up that reaction without actually being used in that reaction themselves. And so instead of that reaction taking thousands of years, it happens very quickly or much faster. And that makes it where we can actually use that reaction in biology to sustain life. Because if our reactions all took hundreds of years to happen, we couldn't do the amount of work that our cells do. And so I talked about how a lot of the times these chemical reactions are spontaneous when they go from a high state of energy to a low state of energy, where we go from potentially a big molecule that has lots of bonds and lots of potential energy into many smaller molecules that have a lower potential energy. And that is spontaneous, but what if we were trying to do, that would be like a catabolic pathway, but what if we were trying to do like an anabolic pathway where we had smaller component substrates and we were trying to build a bigger product, which has much higher energy levels or potential energy than your substrates. And so a lot of the times, for instance, with plants or photosynthetic algae, they'll use the sunlight, the sunlight itself to drive that chemical reaction and provide the energy to push that reaction to that higher energy state. In other instances within the cells, like other bacteria that can't use the sun, or for instance, us that can't use the sun, we can use ATP, which is this high energy molecule called adenosine triphosphate, which has adenosine nucleotide, a nitrogenous base, a ribose sugar, and then 3 phosphate groups, where each of those phosphate groups have a special bond that has a high level of energy. And when we transfer these phosphate groups onto a specific molecule, we're essentially transferring a large amount of energy to that substrate, and we're able to, or to that protein, and we're able to facilitate the work or push that reaction from a low energy state to that high energy state. And ATP is something that is found ubiquitously across life, and it's found in high concentrations because it's being used in many, many different systems across the cells. And so you can think of these catabolic pathways that break things down, and these anabolic pathways as building things up in the terms of this metabolism of maintaining this homeostasis, where we bring in these nutrients, and these nutrients necessarily might not be what we need currently for what we're doing or for the current environment. And so we're going to have to break those nutrients down, those carbohydrates, those lipids, and break them down into their components. And then we excrete byproducts like carbon dioxide and water, but we also then have all of these pieces, these scrap pieces. And these anabolic pathways can then take these scrap pieces and start building specifically what we need for the moment when we need it. It's really neat, but it is quite literally, if you look up like full metabolic pathway or something, it is absolutely insane how many pathways there are, how interconnected they are, and the complexity of the whole thing. And so in most cases, people are not studying the metabolism in its entirety. They're studying very specific pathways or very specific systems within metabolism or specific effects on metabolism. So going back to enzymes and how these enzymes help facilitate these chemical reactions, because they are catalysts. Catalyst is a chemistry term, and it doesn't necessarily have to be enzymes. You can have inorganic catalysts. Excuse me. Sorry. And so we talked about how these enzymes speed up the reactions, and if they didn't speed up these reactions would be incredibly slow and could not be leveraged for biological processes. And that's just, I think that's really neat. And so what's really interesting is each of these, we talked about how metabolism is like thousands of chemical pathways. Wow, wow. But then we also have enzymes that are specifically paired for each of those chemical reactions, that there isn't just one enzyme that just does it all, that in most of the cases there are enzymes that are fairly specific or slightly generic that can be used for each of those reactions. And we'll talk about kind of why that is a little bit later. we're going to talk a little bit more about these chemical reactions and how they operate. So we talked about how there's kind of these energy states where you have this high state of energy and a low state of energy. And if it's going from a high state to a low state, just like water, it's going to flow more spontaneously to that low state of energy. Kind of like how you have this depiction of someone rolling a rock down a hill. Um, the enzyme is helping this rock roll down this hill. But if it did, it wouldn't easily do it by itself. But it could do it by itself, just it would be a lot harder. And so that harder to do, or that like resistance to that like spontaneous like flow of the reaction, is what's called activation energy. And you can kind of think of, if we're looking at like the state of energy in the reaction, going from the beginning to the end of the reaction, you kind of have to kick start, like we talked about a little bit earlier, with either ATP or light or some other chemical energy source. You have to kind of kick start the reaction for it to actually get going. And so enzymes, instead of directly like providing that energy, a lot of the times they're just providing a safe place for this reaction to take place by lowering this activation energy. And so you can kind of think about, if it was like a hill, you could see that the enzyme reduces the height of that hill and makes it where it's a lot easier to overcome and get into that reaction and actually go into our products. And so these enzymes, I mentioned just a couple of seconds ago that these enzymes are specific to a specific substrate or a specific chemical reaction. And most of the time, they're not very generic. And that's because enzymes have a specific shape in 3D space, and that 3D space has a little pocket called an active site where that substrate can bind in. And just like a lock and a key, that enzyme and that substrate is particularly made for each other to and has particularly developed has the enzyme has evolved so that its 3D shape fits that substrate well. And when that substrate gets into that active site, it allows for that chemical reaction to be more favorable for some set of reasons. And it can vary depending on what the enzyme is and what's actually happening with the reaction. But again, just like there's thousands of chemical reactions, there are thousands of enzymes that help those reactions. And so enzymes can sometimes be active and they can be inactive, where sometimes they're in the right shape for that substrate to bind in, and sometimes those substrates can be in not the right shape where they can't bind in. And there are enzymes called apoenzymes, which would be the inactive enzymes. And when these inactive enzymes are bound by a cofactor, when a cofactor or a coenzyme, it creates the right shape for that substrate to bind in. And it converts that apoenzyme to a holoenzyme. Most people don't use these terms. You're going to see them in your textbooks and things, but a lot of the time you're going to see an inactive form and an active form. And so but when these cofactors or cofactors, which would be like a metal ion or a other kind of chemical, usually a metal ion is pretty common, or a coenzyme, which would be like maybe a small peptide or a small other protein that comes in and binds. when they bind, they conjugate with one another and they create a bigger protein that's made up of two separate subunits that create a functional protein. And these 3D structures can be talked about in a few different ways, where you have primary structure, secondary structure, tertiary structure, and quaternary structure, where primary structure is if you are just essentially reading the amino acids, and the amino acids are like chained together like beads on a string, where you just are reading along. You have no idea where they are in 3D space in that folded protein. And that's the primary structure. The secondary structure is when hydrogen bonds start to take hold and start to form these first in these structures called either alpha helices or beta pleated sheets. We'll see pictures of those in a minute. And then eventually those secondary structures come together and those disordered loops come together and they create what's known as a tertiary structure, which is that fully formed, fully folded protein. And those are held together by van der Waals forces, disulfide bridges, and hydrogen bonds. And then you have quaternary structures if you have this protein comes together with another set of proteins. So, for instance, you might have a coenzyme that comes together, and then that creates this quaternary structure where you have one subunit and a second subunit, and this quaternary structure between the two. And so this 3D shape in this binding pocket, or what's called the active site, is really important for the substrate to actually bind. And if that if that binding pocket isn't formed right, or the protein isn't folded right, that substrate isn't going to be able to bind, and potentially you wouldn't be able to help along that chemical reaction, which could lead to disease or disorder or dysfunction within the cell. And so here's a visualization of the different amino acids, the structures of proteins, where you have primary structure where it's just the string of amino acids and you have no idea where it is in that final protein structure. You then have hydrogen bonds that form between these amino acids, and they can form what's known as an alpha helices, which are these tight coils, or these beta pleated sheets, which are these alternate arranged strands of amino acids. And these amino acids, not that you need to know, but these amino acids have like a backbone, and then they have like a radical group or the amino group. And in these pleated sheets, they alternate back and forth on which direction or which side they stick out on the sheet. And so that's kind of why you have that pleated pattern. You also see, if you want to know where pleated, the word comes, like the pleated, you might see the word pleated in fabrics where you have tight folds that are reoccurring. But anyway, these structures are held together by hydrogen bonds, and they are some of the first structures that form. And then you also have these intrinsically disordered regions, or these loops that connect them. And so then, I think I'm all the way out. And then these loops come together and start to form hydrogen bonds. They start forming hydrogen bonds and disulfide covalent bonds between sulfur atoms that create this tertiary structure, or this final 3D shape. And then if it comes together with other proteins, it will have that quaternary structure. And so if you look here, this is a crystal structure of what a protein looks like. And you can see the alpha helices in there. And there is a pleated sheet right here. It's kind of hard to see. There's a little one right there. It's hard to see. But there's a lot of these intrinsically disordered loops that don't really have a defined secondary structure, but they have this defined 3D fold. And if you looked at this protein in all of your different instances in different cells or different or in the same cell or in different cells or in different organisms, it'll be folded fairly similarly, if not the exact same. Oops, didn't know you could do that. And so you can think of these proteins in two different ways, where you have either a lock and key model. Sorry, not, yes, a lock and key model, but you can either think of these proteins as rigid bodies, or you can think of them as kind of flexible, kind of breathing proteins. And so the rigid body model is a really nice way of thinking of these proteins because it's easy to do like with simulations and things because they don't move very much. And what it thinks is that the protein is folded and it is what it is. It is pretty much glued and stationary in place. And it has that active site that the substrate comes in just like a lock and key. And if it doesn't fit in there, it can't be catalyzed by the enzyme. Okay. Like this one isn't the right size and so right shape, and so it can't fit in that active site, so it doesn't work. But a more nuanced and a more accurate view is that this, just like all of your like all molecules, when they move around. And so this is just a string of molecules that are all folded up in a very neat and orderly fashion. So they're all going to move a little bit. And so they kind of all have this kind of breathing to them. And what that means is even in the active site, there's some intrinsic movement that happens. And so maybe the substrate doesn't always fit perfectly in that active site. And that movement and that intrinsic movement might allow for it to fit. And so this is what's called as an induced fit model. And and this is a more accurate view of what's happening. Something also we'll talk about later is how this intrinsic movement kind of allows for regulation of this active site and allows for chemical reactions to not take place by interacting with a different region. And so I mentioned earlier that you can have functional enzymes and you can have infunctional enzymes. And sometimes these can be very deliberate, where enzymes can be used either outside of the cell and they can be called exoenzymes, or they could be inside the cell and be called endoenzymes. And when they're needing to be used in one place or the other, they'll probably be inactive in one. Usually you don't have an inactive form on your endoenzymes, but for your exoenzymes, whatever you're doing outside the cell, you don't want to be doing that inside the cell. And so you have a particular way to make it where this enzyme is inactive. And you can think about this in a few different ways. So we know that this enzyme acts on a substrate and does a chemical reaction. It does that by taking that substrate and binding it into an active site. And so how can we make it where this enzyme isn't active? Well, it has to be properly folded. So we could make it where it's not folded properly until it's outside of the cell, and that happens. We could physically cover the enzyme, that we can physically cover that active site. Make it where that substrate can't even get in, and that happens. And we have to cleave that away before it becomes active. And you would want to do that outside of the cell for it to do that. And that happens. Let's see. You could have a substrate that's like a fake substrate that could fit in that active site and prevent the chemical reaction that you don't want to happen. And if it fits in that substrate, maybe it stops it. That I don't know if that happens off the top of my head, but it does happen with drugs. That's how drugs inactivate enzymes. And so the cells have these very intuitive ways of controlling and regulating when and where these proteins are expressed and when they're active. And so sometimes you need an enzyme all of the time. And that's what's called a constitutive enzyme. And these enzymes are really important for just like daily life, and they're needed all of the time. In research, these enzymes are really important as standards to normalize like your experiments to. And so like say you have some sets of cells and you measure a protein of interest in each of them. And you're like, okay, it's really high here, low here, and high here. So something in this middle one is of something's weird that's causing my protein of interest to be really low. It's probably XYZ condition, I hypothesize. But if we look at these constitutive enzymes, they are usually expressed at the same level in every cell. Sometimes they're the same level in different cells, like different cell types. Sometimes they're the same level in different organisms. Sometimes they're like, that's how important it is for life, and it has to be made at a specific level. But anyway, in this case, we're just saying it's used and we know that it's uniform in all of these cells. And so if we measure that and we see, again, it's high, low, and high, but they should be the same. Well, that middle sample, which had low constitutive enzymes, something's wrong with that sample, and my result is flawed. There's something wrong with it. There's an error. And so that's why they're really helpful in molecular biology. Excuse me. And in research, and if you look in publications that use like Western blots and things, this is very common. Even in like RNA, looking at RNA levels, RNA-seqs, they use constitutive enzymes levels all the time. Regulated enzymes, I'm going to move on now. Regulated enzymes, on the other hand, are something that you need at a specific time in development, but not all the time. And so you have to have a system to say, we need it, we don't need it. We need to make it, we need to degrade it. And then also, we need to not express it. Oh, I can't, you're not seeing my hands. And you need to not express it at these other times. We only need to express it here. We only need to build it here. We need to degrade it. And those are what are called regulated enzymes. And there are a lot of these. And so enzymes, we're going to talk about condensation reactions real quick. And so enzymes can either like do like phosphorylation, or they can do what's known as condensation. And we're going to talk about how it takes the water from two substrates, pulls it out, and forms a bond between them. And so we're taking two small components, taking out water and building a bigger molecule. And so this is an anabolic reaction. We're taking smaller components and building something bigger. And in the process, we're producing water. And that water came from those two components to begin with. On the other hand, the reverse of this is we're taking a big product that has an OH. Oh, sorry, it does backwards. That doesn't have an OH. We add in a water and we break it into its component substrates, into two component products. Sorry, we take one big substrate and break it up into two component products, smaller products. by consuming the water. And this is what's known as a hydrolysis reaction. And so you can think of the name of water, of hydro, and lysis, which means to break. And so water breaking reaction. And so we're taking a water and we're breaking up this molecule into component pieces. And condensation, you can think of we are removing the water and we're putting the water into the environment. Condensation, you have if this got water on the outside, it would be condensation. It's getting water on the outside of the environment. So you can think of we're producing a water, we're pushing a water out. And you'll have to just kind of memorize that it is a anabolic building reaction. Excuse me. Sorry about that. Okay, so we talked about how these proteins are these folded chains of amino acids, and that they're molecules just like any other, and that they move and they're dynamic and they kind of breathe and they just kind of have this intrinsic movement about them. And just like all molecules, uh, physiological conditions like temperature, pH, or osmotic pressure affect how these molecules move around. And so you can think about when you have a molecule, we're going to kind of put away the protein for a second. We have a molecule, we have a temperature, and we increase that temperature, that molecule moves more and more. And it moves a lot. And it moves at a random Brownian motion kind of way. Now, if we had two, both of them are moving in kind of a Brownian motion kind of way, but they can't kind of do that because they're connected. And so you can think as we increase the temperature with a folded protein, it's just a chain of amino acids similar to that, all kind of doing that, all connected, all interacting with each other and bouncing into one another, right? And so when we increase that temperature, there's going to be a more intrinsic movement in that protein, and potentially it could start to be unfolded in what's called denature. You can also have pH have an impact, and pH is a measure of like the concentration of hydrogen ions, and it's the log concentration of hydrogen ions in a solution where one has incredibly high levels of hydrogen ions and 14 has incredibly low levels of hydrogen ions. And these proteins are being held together by a few covalent bonds, disulfide bridges, a few van der Waals, like in van der Waals forces, there's probably a lot of van der Waals forces, and hydrogen bonds. Now, the hydrogen bonds are disrupted when there's hydrogen ions in a solution. And so if you're starting to break these hydrogen bonds that are holding these folds together in specific shapes, again, you're going to denature your protein. With osmotic pressure, on the other hand, you can think about osmotic pressure as like the general density of a solvent, a solute, and water's movement towards diluting that solute, because water wants everything to be very homogenized and the same. And so that pressure can change the movements of a molecule, so kind of like a river kind of changes your movement when you're swimming in it. Also, the density of those molecules affects how things move and interact, because these molecules are moving, they're bouncing into one another. And if there's a high density, it's going to bounce into each other more often. It's going to not be able to move as much. So you have to think about this in like a physical kind of reality as well, is not just, oh, these things kind of happen on paper, but you have to think about how the physical environment are affecting these reactions that we're looking and reading and learning about in the book. So these proteins, we talked about how there's kind of, you can have like drugs that inactivate substrates, and you can have drugs that act as inhibitors inside the active site itself, just that completely you can't be in here because I'm in here, nana nana boo boo. Or you can have non-competitive inhibition, which is where something binds to somewhere other than the active site, and that somehow makes a change in that protein that doesn't allow for that substrate to get into that active site, okay? And so that's what's called non-competitive inhibition or allosteric inhibition. And so here we can see a diagram of the two where typically you have the normal substrate that fits into this enzyme and it can be processed into the products. Otherwise, you have this drug or this compound. It doesn't have to be a drug that's like been designed. It can just be any molecule that's similarly shaped to the substrate. It can get in there and it can actually block that substrate from getting in and slow down this reaction. And depending on how well it binds, sometimes it binds irreversibly, where it pretty much is like that protein is out of commission, doesn't do anything anymore. And that's an irreversible, that would be considered irreversible. But a lot of the time, just being able to bind and unbind is enough to make it where it's harder to do this reaction and slows that reaction down. And so you can kind of think about this process of like, if you had, let's think of a carnival game and you have like this little hole and you're throwing ping pongs through this hole, and ever so often you get it through that hole. And that's like the substrate getting into that active site and being processed into the products. Now you're doing it, you're getting the hang of it, you're going quick. Eventually, some other guy comes up and starts throwing their balls that are a different color through the hole. And sometimes it hits your ball and knocks it through. And you're like, I would have got into that hole if you didn't throw yours there. Right. And so that's kind of what's happening with this competitive inhibition, is because they're just there and they're even trying to get into that active site and they're getting in and they're blocking us. It's making if we were taking like a score, it's making my score worse. And that's exactly what's happening with this competitive inhibition. On the other hand, allosteric inhibition, which is non-competitive, you have the substrate able to be processed into its reactants, but maybe you have this second site that something binds to that like closes off the active site or blocks it in some way. And this is what's called allosteric inhibition. And this is usually it binds and it creates some kind of fold change, a change in the fold of the protein, and it blocks the substrate binding, or it blocks like the reaction to happen. And so a lot of the times you'll see this in the into these metabolic pathways, that the products of this enzyme actually bind into that secondary pocket and inhibit it. And you can think about what would kind of happen with this. So if this protein is going through, it's accumulating products and it's just pumping out products, you just piling up just more and more products. Eventually, you don't need that many. Eventually, this is going to be bad for you, probably, like, right? And so the cell wants to regulate how much can pile up at one time. And so when it starts to pile up, it's going to start becoming more likely that these products encounter this secondary site and bind to it. And they're going to bind to it more often the more that there are these products about. And so when these products pile up, the more of these enzymes that are going to be shut down. And this is a regulatory mechanism that shuts down this pathway to not allow products to overaccumulate in the cell. And this is a type of feedback regulation. There's positive feedback regulation and negative feedback regulation, but we'll talk about those probably in another chapter. We talked about how chemical reactions sometimes go from high energy to low energy, and sometimes they go from low energy to high energy, and sometimes they need when they go from low energy to high energy, they need something to drive them, and they need to consume energy, and those are called endergonic reactions. When they're spontaneous and they go from a high energy state to a low energy state and spontaneously happen, those are releasing energy, and those are exergonic reactions. And typically, if you had like instead of like a biological reaction, if you just think of like a hand warmer, there's a reaction that happens there, and it's releasing heat, and that's an that's an exergonic reaction where it's releasing the heat. If that heat warmer was rechargeable, that recharging process would be an endergonic reaction where it's consuming energy to get to its new state. So anyway, here's a good diagram of that where exergonic is going from a substrate and releasing energy. This would be something from like a high energy state to a low energy state and releasing energy along the way. And then you have endergonic, where you have energy coming in to help drive the substrates to that higher energy product. And so cellular respiration is a process of exergonic reaction. Exergonic is where we're releasing energy. And then we're going to capture that energy in ATP and high energy carrier molecules through a series of reactions. And before we talk about exactly what those are, we're going to talk about what redox reactions are. And you can think about oil rig, where oil rig. Sorry, I don't write with the mouse very well. Where we have reduction and oxidation, where reduction is where our substrate is gaining electrons, and our oxidized is losing electrons. And if you think of the acronym oil rig, oxidative is losing electrons, and reduction is gaining electrons. And so a lot of the times these happen in one specific reaction, and it is a way, let me see how I can clear that off. Oh, I don't think I have to clear it off. Give me a sec. I'll figure it out. Maybe. Here, we'll do this. Discard. Ta-da. Okay. So these oxidation and reduction reactions, or redox reactions, happen in the same chemical reaction, where one of them is where one of the substrates is oxidized and one of the substrates is reduced, where one is oil rig, oil rig. Sorry, I'm having functional issues today. So oil rig, oxidation is loss. And so we lose electrons here. And rig, reduction, is gained, where this substrate is gaining electrons. So now back to respiration, we have glucose, which is the 6 carbon sugar, which goes from a high energy state to a low energy state and to carbon dioxide and water. And in that process, we release the hydrogen ions and electrons, and we try to capture the energy that's released as we move from this high energy to low energy state that's released into the environment. And we do this by trying to eventually capture it into what is called adenosine triphosphate, or ATP, which, like I said, has this nitrogenous base and then ribose sugar. And then we have the phosphate group. And if you look at just this first phosphate, this is what would be used for a RNA backbone. But then you have these other 2 phosphate groups, terminal phosphate, these 2 phosphate groups, and then this terminal phosphate group. This terminal phosphate group has an incredibly high energy bond attached to it that we use, and we break that bond, and we transfer this phosphate group onto substrates to transfer that energy and allow for us to use that energy to drive the chemical reaction. And so we can form ATP in a few different ways. We can actually form it onto, we can actually take the phosphate from a molecule and put it onto ATP with an enzyme just directly. And that's called phosphate level phosphorylation. And through the pathways that we learn, we're going to see a couple of instances of this. And then we have oxidative phosphorylation, and this is the process that we will see in bacteria on the cell membrane or that inner membrane, where we use an ATP synthase, or just this motor protein that's really cool that just pumps out a high level of ATP, and at that place we do oxidative phosphorylation. And then in photosynthetic organisms, they can do photophosphorylation, where they use light to help provide the energy to transfer phosphates onto what's a DP, which would be adenosine diphosphate. So I'm going to go over that real quick. If you look at this, this is adenosine triphosphate because there's 3 phosphates. It would be diphosphate. If there's 2 phosphates, it would be monophosphate if it's 1 phosphate. If you can see that labeling here. And so we're going to now talk about respiration and how we break down these large molecules into smaller molecules and capture the energy in that. And so these respiratory pathways can kind of have 2 different flavors, where you have aerobic respiration or you can have anaerobic respiration. Aerobic would be with oxygen, and anaerobic, would be without, so without oxygen. And what this is referring to is at the very end of this process, we're kind of using electrons a lot, and there's going to be a set of electrons that need to be bound to something. And so we have what's called a final electron acceptor. And typically within aerobic respiration, we use all the time. In aerobic respiration, we use oxygen. And in anaerobic respiration, we use inorganic non-oxygen molecules, like different gases. Woo! Like nitrate. Right. I forgot what SO4 is. Let's see. It's sulfate. It's a sulfate ion. And so you'll have these chemical groups on different molecules, and these groups will act as those final electron acceptors, and they can vary depending on the organism. And then you have fermentation, which is kind of a partial glycolysis that creates different organic alcohols and acids. And so for aerobic respiration and anaerobic respiration, you have these 3 steps of glycolysis, which breaks down glucose into pyruvate. You then have the Krebs cycle, which breaks it down further and harvests these high energy molecules. And then you have the electron transport chain, which harvests all of these high energy molecules to make a large number of ATP. And so this is kind of what that looks like. It can drawn out as a chemical equation where you have the adenosine diphosphate and then the inorganic phosphate groups. Sorry, that's an organic phosphate group. My bad. Woo. And then they woo go through aerobic respiration, breaking down that glucose, harvesting that energy, and finally making 38 ATP, which is a large number of ATP molecules. This is in an ideal world. And so not always will one glucose molecule make 38 ATP. So we're going to talk about glycolysis, where we have this, I'm going to move my thing around again, where we have this 6-carbon sugar, which the first step is getting phosphorylated. And this takes an ATP to phosphorylate this, and so it takes work to get this phosphate group on this glucose. And so now we have glucose 6-phosphate, and this glucose 6-phosphate then gets rearranged and into fructose 6-phosphate, and another ATP is provided to phosphorylate the first carbon. And now we have fructose 1,6-bisphosphate because we have 2 phosphate groups. Notice that we've invested 2 ATP molecules into this pathway. So we're -2 energy molecules. Then at this step, we break this fructose 1,6-bisphosphate into 2 component molecules that are kind of mirrors of one another that can kind of be isomerized or rearranged into one another. And so now we'll have eventually they'll both become glyceraldehyde 3-phosphate. And there'll be 2 of these molecules because we've broken this from 1 into 2, and then this gets converted here. So we'll have 2 of them. And what we're able to do is we're able to harvest a high energy carrier molecule, or NAD to NADH, and convert it into NADH, or charge it with electrons. And also we're able to phosphorylate these 2 substrates. We're able to make 2 of these carrier molecules because we have 2 of these glyceraldehydes. Then we're able to transfer this phosphate group onto 2 ATPs, and we're able to generate 2 ATPs. And so now throughout this whole process, we've invested 2 ATPs, we've gained 2 ATPs. And through another series of steps, we rearrange the phosphate and generate another set. I'm going to move my Zoom control panel again. And we rearrange the phosphate group, and then we're able to then harvest 2 more ATPs. And what this creates is 2 pyruvic acids. And so we've been able to invest 2 ATPs. We've been able to net 4 ATPs. Because we have 2 here, which makes this a neutral, net 0, and then 2, this makes us +2. So we have +2, boop, 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 boop. We got +2, we got +2, we got +2. What are you gonna do about it, bitch? I'm so sorry. Woo. Anyway, wow. Sorry, I have functional movement disorder. And so my brain kind of forgets how to function sometimes. So it was just really exciting. We've gained a net 2 ATP molecules through this process of breaking glucose into 2 pyruvates. This process of glycolysis breaks down it into 2 pyruvates and nets us 2 ATPs. Just as I'm reiterating to help reiterate. Oh, here we go. We're reiterating it again. We have glucose and we've generated 2 ATPs net, and we also generated these carrier molecules that we're going to use in the electron transport chain. Then we're going to have these pyruvic acids get shuttled into the Krebs cycle. First, they have to be conjugated onto coenzyme A, or CoA, and then they'll be pushed into the citric acid cycle. And so here's the process of this preparation, where pyruvic acid is is converted and conjugated into coenzyme is converted into acetyl-CoA by conjugating it to coenzyme A and taking off a carbon dioxide. It also is able to generate another carrier molecule. And then we enter into the Krebs cycle once we have it conjugated, right? And it goes through and it takes this acetyl-CoA and binds it to this oxaloacetate, which is the leftover of the Krebs cycle. And it comes in and it gets converted into citrate once it's bound in. Once it is citrate, it gets rearranged into isocitrate. Then two carrier molecules are generated, and it becomes alpha-ketoglutarate. I know this is really hard, but all we're wanting to know is that these carrier molecules are being generated through this process of slowly chipping away at this molecule. But we couldn't just chip away at this molecule as it was as pyruvic acid. We first kind of had to prepare it to go through this cyclical pathway. And we had to do that first by making it acetyl-CoA and then conjugating it with the leftover oxaloacetate. And so once we're at alpha-ketoglutarate, we get another set of binding molecules. Sorry, we get another set of energy carrying molecules, which are NADH. And we generate succinyl-CoA. We're able to then harvest an ATP molecule, just one. I was thinking of glycolysis when I was thinking too. We harvest one ATP molecule and we're able to generate succinate and then finally generate a new carrier molecule. This one we haven't seen before. This one's called FAD or FADH2. And FAD is a separate carrier molecule than NAD, but both of them are going to be used in the electron transport chain. Notice that NADH is carrying one hydrogen ion, while FAD has two hydrogen atoms. I always said ion, I meant atoms. So from succinate, that gets transferred into fruminate, and then fruminate into malate by adding water, then malate into oxaloacetate by generating another carrier molecule. And again, don't really need to know all of these components. What you need to know is that through this process, we prepare these pyruvic acids, and we're generating these these carrier molecules. I accidentally skipped over this part, but this is where our carbon dioxide comes from that we exhale and breathe out when we go through cellular respiration. You then generate an ATP molecule and another carrier molecule. These carrier molecules are then taken to what's called the electron transport system. And in us, we use mitochondria, but in prokaryotes, we don't have membrane-bound organelles typically. And so prokaryotes are going to be using the cell membrane, and they're going to use that inner membrane that's inside of that cell wall. That's right next to the cytoplasm. And then there's the cell membrane, and then there's the periplasmic space, and then there's the peptidoglycan cell wall. They're going to use that cell membrane there for this ETS system or electron transport system. And here's a picture of it after I just described it really well. So here's that inner membrane or that cell membrane, that periplasmic space, and that cell wall made of peptidoglycan. And what we're going to be doing is eventually bringing in those carrier molecules and pumping in those hydrogen ions, that hydrogen atoms as protons into the periplasmic space to create a chemical gradient that we can then leverage to do work. And we do the same thing in mitochondria, where we shuttle in these high energy molecules, the same ones, and pump these hydrogens into what's known as the inner membrane space. But this is kind of confusing because this is only in eukaryotes. But these hydrogen ions or protons create this like chemical gradient. And this chemical gradient allows us to pump that kind of allows us to use what kind of like a water wheel or like a motor to take in ADP and phosphate and generate ATP by using the energy from the movement of these protons through the membrane. But this only can occur if we can have this chemical gradient occur. And what's named as chemoacidosis. No, that's wrong. Chemoosmosis, where there's this gradient where the protons want to have the same density of ions on both sides. The protons want to be at the same concentration in this space as this space. And so there's this pressure or this proton motive force that drives these protons through the membrane. And we can leverage this proton motive force to do work. And we use a ATP synthase to do this. And this ATP synthase is this mechanism, this rotary motor that cycles, that kind of like as the hydrogen protons move through here, it turns this and it like pushes these together and it makes the ATP. It's really neat. I might find a video, an animation of this to show you guys later. Not now. And so, again, we're taking, through this process, we're taking this, actually, it's what I mean. Yeah, let's go a little bit more into this a little bit. So these protons are being pumped into this space. And what's happening is just like hydrogens are made of one proton and one electron. And so if we're moving protons over here, where are those electrons going? Those electrons are moving through a system of protein complexes that are inside and embedded in the membrane. And this happens in both prokaryotes and in eukaryotes. And I'm going to look at this one because the electron transport system is what we're really interested in here, where these protons are getting pumped through the membrane, and then the electrons are being captured by these proteins and being pushed inside this. And so there's this positive charge here and a negative charge here. So there's the positive charge and then the negative charge. And so that is reconciled once those protons get pumped into the cytoplasm again. But that's kind of what's actually happening if you want to think of it a little bit more holistically. So anyway, all the way, we have glucose, which is broken into pyruvic acid, which takes a net of 2 ATP. Again, this wasn't just spontaneous. It needed some energy. We had to put in some ATP to make this process happen. We took glucose, put in some ATP to get this pyruvic acid and ATP out and this carrier molecule. It didn't just happen, but we did come out ahead at least. And then you have this transition step that is the preparation of pyruvic acid into acetyl-CoA by conjugating it with the coenzyme A and taking off this carbon dioxide and carrier molecules, hydrogen atom. Then you enter into the Krebs cycle that goes through twice because we have 2 pyruvic acids. And it generates the carbon dioxide that we exhale, and it generates carrier molecules and ATP as a product. It then shuttles all of these electron carrier molecules to the cell membrane, to the electron transport system for oxidative phosphorylation, which is the process of pushing the electron transport system is pushing those protons and making that chemical gradient. And then oxidative phosphorylation is the process of phosphorylating and generating all of those ATPs. And so eventually, those electrons have to be, I think I said something wrong earlier. And so that final, when I talked about those electrons then coming back together with those protons, that's not exactly true. They kind of are, but like, yeah, they are coming together with oxygen. And that's why oxygen is that final electron acceptor. We essentially stole the protons and left us with this electron. And if we didn't have something to take up that electron, it would cause harm in our body. And so we have this oxygen that takes up this electron as the final electron acceptor. And that's aerobic respiration. That's with oxygen. But if we don't have oxygen, what are we going to do about this electron? And we can use reactive groups like nitrate and nitrite, or sulfate to capture those final electrons. And then when we look at fermentation, it's kind of this glucose process, this glycolysis process, but instead of breaking it down into pyruvic acid, we're breaking it down into organic acids and organic alcohols and gases instead of pyruvic acid. And so, for instance, we have lactic acid or alcohol or formaldehyde, and something that is of, and so I'll talk about that in a little bit. So you can see how this glucose is creating pyruvic acid and then breaking it down further. Yeah, sorry, I said that wrong. You can see how this glucose is breaking down the pyruvate and breaking it down into further components like different organic alcohols and acids. I said instead of pyruvic acid, that's wrong. It goes to pyruvic acid and then it branches off into all of these different things. And depending on the species, it branches off into all of these different things. Like, so for instance, lactobacillus can make lactic acid like you would want for yogurt, or you could have yeast that takes pyruvic acid and breaks it down into ethanol. Or for instance, Enterobacter can make 2,3-butadiol. I don't know what that is, but you can make it with Enterobacter. And so something that's really interesting, and that's the conclusion of chapter 8, but something that's really interesting about this process is that all of the steps in here, all of the compounds that are made throughout this process have these secondary metabolic pathways that generate very necessary compounds like amino acids, signaling compounds, that are important for life also. And so these processes, like I said, are incredibly connected and interconnected, and they feed off of each other. And so this process doesn't just make energy, but it also is feeding in making all of these necessary molecules that are necessary for life. And so with that, that is our lecture. And so I apologize for my functional issues. I hope it wasn't too bad, and have a great rest of your week. Bye-bye.